Hi students, hope you all are fine, no? So on my previous class, I had explained about the alimentary canal and digestive glands. Today I am explaining about the physiology of digestion. So physiology of digestion, before going to the physiology of digestion, you should know what is digestion. The mammalian food consists of protein, lipid, carbohydrate, minerals, water and vitamins. Minerals, water and vitamins can be directly absorbed by the gastrointestinal tract, but due to their large size, protein, carbohydrate and lipid cannot be absorbed directly inside our gastrointestinal tract. They have to be broken down into their simpler form and then they are absorbed inside the gastrointestinal tract. So digestion is the breakdown of complex food particles into their simpler form. Okay? And now you focus on the diagram of the digestive system. So this is buccal cavity. After ingestion of food, the three pairs of salivary glands secrete saliva, and saliva contains salivary amylase, which is a carbohydrate digestive enzyme. So digestion starts inside our buccal cavity. Okay, so inside our buccal cavity, salivary amylase acts on the starch part of the food, and starch is broken down into maltose. So starch is broken down into maltose in the presence of salivary amylase. Salivary amylase and water. So after digestion of starch, the food mixture inside the buccal cavity is known as bolus. And the saliva makes the food mixture slippery, and the food mixture moves down through the esophagus into the stomach. So just after entering inside the stomach, the gastric glands secrete hydrochloric acid and digestive enzymes. And gastric glands consist of three types of cells: perital cell, Schiff cell and mucus cell, as I had told you on my previous class. So perital cell secretes hydrochloric acid, and hydrochloric acid stops the action of salivary amylase inside the stomach. And Schiff cell secretes two types of enzymes: proteolytic enzyme and lipolytic enzyme. Proteolytic enzyme means protein digestive enzyme, pepsinogen, and lipolytic enzyme means gastric lipase, lipid digestive enzyme. Okay? The proteolytic enzyme is secreted in their inactive form, pepsinogen. So pepsinogen is converted into their active form, pepsin, in the presence of hydrochloric acid. So pepsinogen is converted into their active form, pepsin, in the presence of hydrochloric acid. And pepsin action, the protein part of our food inside the stomach, and the protein is broken down into peptones, proteases and polypeptide, because protein cannot be broken down directly into their simpler form amino acid. First, they have to broken down into peptones, proteases and polypeptide inside the stomach. And then gastric lipase acts on the lipid part of the food mixture like this. Lipid is broken down into fatty acid and glycerol. A glycerol in the presence of gastric lipase. Okay, so you should always remember that inside the stomach no carbohydrate digestion occurs because gastric glands do not secrete carbohydrate digestive enzyme. And now the semi-digested food mixture inside the stomach is known as chyme. And chyme enters into the duodenal part of the small intestine through the pyloric sphincter of the pyloric stomach. And now the food mixture is mixed with the bile. Bile is secreted by the liver. So liver secretes bile, which is drained out through the bile duct and temporarily reserved inside the gallbladder. And from the gallbladder, the bile is drained into the duodenal part of the small intestine. And the digestion inside the stomach activates by a hormone known as gastrin. So likewise, in the duodenum part, a hormone called cholecystokinin or CCK activates the gallbladder to squeeze out the bile. And now the food mixture is mixed with the bile, and bile has bile salt and bile pigment. So bile salt is responsible for the emulsification of fats in the food mixture. Emulsification means breaking down of fat molecule into some fine droplets. So after mixing with the bile, the food mixture now is mixed with the pancreatic juice, which is secreted by the pancreas. So pancreatic juice contains three types of enzymes. Proteolytic enzymes, that means protein digestive enzyme. Lipolytic enzyme, that means lipid digestive enzyme. And amylolytic enzyme, that means carbohydrate digestive enzyme. So proteolytic enzyme is trypsin, which is secreted in their inactive form, trypsinogen. So trypsinogen is converted into their active form. Trypsinogen is converted into their active form trypsin. Trypsin in the presence of enteropeptidase. So the inactive form of trypsinogen is converted into their active form trypsin in the presence of an enzyme enteropeptidase. And trypsin acts on the protein part. That means trypsin acts on peptones, proteases and polypeptide. And peptones, proteases and polypeptide is converted into tripeptide and dipeptide. So after breaking down of peptones, proteases and polypeptide into tri and dipeptide, now the amylolytic enzyme, which is known as amylopsin, amylopsin acts on the remaining part of the starch. And starch is broken down into maltose in the presence of amylopsin. And then the lipolytic enzyme of pancreatic juice is known as stepsin. So stepsin acts on the remaining part of the fats, and fats is broken down into fatty acid and glycerol. And then the food mixture is mixed with the intestinal juices. So intestinal juices contains 3 types of enzymes. Number 1, ellipsin, which is a protein digestive enzyme, and sucrose digestive enzyme, lactose digestive enzyme, and lipid digestive enzyme, enteric lipase. So ellipsin acts on tripeptide and dipeptide, and tri plus dipeptide, and tripeptide and dipeptide is converted into amino acid, which can be absorbed inside our gastrointestinal tract. And sucrase acts on sucrose, and sucrose is broken down into glucose plus fructose. Lactase acts on lactose, and lactose is broken down into glucose plus galactose. And maltase acts on maltose, and maltose is broken down into glucose plus glucose. And then enteric lipase acts on the remaining part of the fats, and fats are broken down into fatty acid and glycerol. So digestion is completed inside the small intestine with the help of intestinal juices. And after completion of the digestion, the undigested food particles or waste materials enters into the large intestine. So the colon of the large inside the colon of the large intestine, absorption of water, minerals and sodium occurs, and also facial matters are formed inside the colon of the large intestine. And rectum is the temporary reservoir of feces, and feces are flushed out through this anus. So this is all about the physiology of digestion. Hope you all understood. Okay, thank you.